chapter 13.4 and 14.5, and it's going to be in preparation for our electrophoresis lab, and in talking a little bit about genetic mutations, something called point mutations. So kind of cleaning up here the end of 13.4, which you saved to do right before electrophoresis lab, and 14.5, about genetic mutations, kind of after we talked about transcription and translation. So something that's very important in genetics nowadays, it's something that has a lot of controversy surrounding it, but also a huge potential to change our world for the better, is something called genetic engineering. And genetic engineering is any time we manipulate the DNA of an organism for a specific goal or for a specific purpose. So, for example, manipulating the genome of cattle to have them produce more milk, or plants to make them pesticide resistant, or anything like, or bacteria to produce a specific chemical that we want. All of that is examples of genetic engineering, right? And one of the foundational techniques of genetic engineering is something called gene cloning. And in gene cloning, what we do is take a specific gene that we're interested in because it makes a protein we want or something like that, and we replicate it many times, make many exact clones of it, so we can harvest the protein out. And to do this, what we need is something that can reproduce really quickly, so that we can get a lot of copies of that specific gene. And that's why we use bacteria as what we call vectors. So they carry the gene that we're interested in, and they reproduce many times to hopefully give us many copies of that gene. So what we need to do is we take a gene that we're interested in from a different cell. So we find some cell that has a gene that we're interested in, and we cut that gene out of its DNA, and we insert it into the circular plasmid of the bacteria. And then we put that plasmid back into the bacteria, and we grow them, we culture them. And after they reproduce for however many generations, we can harvest the protein from the bacteria, because the bacteria transcribed and translated that gene into protein into its inside of it. We can harvest that and use it to for various research applications, or various environmental or industrial applications. So gene cloning is extremely important for our genetic engineering potential as a society. Something that's really important, or that allows that to happen, that allows for gene cloning to happen, is being able to cut DNA at a specific place. And we use things called restriction enzymes to do that. To genetically engineer something, or to clone a gene, scientists need to cut out pieces of DNA predictably. So they have to know where exactly it's going to be cut out at. To accomplish this, they use something called restriction enzymes. And these are different enzymes that cut DNA at specific nucleotide sequences. So each restriction enzyme, there are many of them, cuts DNA at one specific sequence. So for example, each restriction enzyme has something that's known as a restriction site, or a given sequence of nucleotides in DNA that it looks for and acts upon. So, for example, this restriction enzyme will look for any sequence of GAATTC, and that's its restriction site. It's where that is active at. And in that sequence, between each G and A, so right here and right here, that restriction enzyme will cut DNA. So the restriction enzyme looks for its restriction site, and then cleaves or cuts DNA at a specific point in that restriction site. And what you get is pieces of DNA that look like this. And some restriction enzymes cut kind of staggered like this. That's what we call sticky ends. And sticky ends are really what's useful in genetic engineering because we can piece these back together in the plasmid to form a whole plasmid. So then what we do is take a fragment. When we cut, maybe this is the plasmid, we cut the plasmid with this restriction enzyme. And then we cut around our specific gene in the other organism with that same restriction enzyme. So we can just piece them back together and create a whole plasmid. And we ligate it together with DNA ligase, and we get a whole recombinant DNA molecule because we recombine the DNA with some other DNA that we've restricted or cut out of another organism we put into our plasmid. So restriction enzymes really help us with gene cloning by being able to cut out a specific sequence with a specific gene and put it in a plasmid. Another thing restriction enzymes are good for are electrophoresis. So scientists can use restriction enzymes to cut up all of someone's DNA into smaller pieces. So since a given restriction enzyme will only cut at a specific sequence, if two individuals like me and you have our DNA cut with this restriction enzyme, that restriction enzyme will create different sized and numbers of pieces because my DNA sequence isn't the same as yours. I might have more of whatever specific DNA sequence, genetic nucleotide sequence that restriction enzyme acts on than you, and then my DNA would be cut into more and smaller pieces than yours. And so we can create a visual comparison between two individuals' DNA and the relative number and size of the DNA fragments created by subjecting both of our DNA to the same restriction enzyme in gel electrophoresis. So what we basically do in gel electrophoresis is we cut samples of DNA and we put them into wells or holes made in a sheet or a slab of agarose gel. So we have a large piece of agarose gel, it's a slab, and we have these little rectangular wells that we can dispense different samples of DNA to be cut by restriction enzymes. And we fill this chamber with the slab in it up with an electrolytic buffer solution, and we pass an electric current through the solution and through the gel. And since DNA is positively charged, the DNA will be attracted to the cathode section, or the positive side of the chamber, and it will start to move down this gel. Now, the larger the DNA fragments are that were created by the restriction enzyme, the slower they'll move. It takes more time to move those bigger, bulkier pieces through the gel, and the smaller pieces will be moved quicker. So if we subject our gel to electricity for a certain amount of time and stop it, we'll get a banding pattern where the shorter sequences will be farther along the gel and the bigger sequences or longer bands will be closer to the wells. And we can visually compare two individuals' banding patterns to see just how similar their DNA is. The more similar they are, the more similar their DNA is. And we can use this technique in crime scene investigation. So if we have a sample of DNA from a crime scene, we can put it in one well, and we can put samples from each suspect in the different wells. Whichever suspect's banding pattern matches that of the crime scene DNA must be the perpetrator. These DNAs must be the same if they match. Second thing we're going to talk about is genetic mutations, or point mutations. So these are mutations that happen by changing a single nucleotide in our DNA strand, changing only one or two nucleotides. So when we talked about genetic mutations, dominant and recessive genetic disorders, those might have to do with whole genes being different. Chromosomal abnormalities are problems with our whole chromosome. These point mutations are just changing one or two individual nucleotides, and they can indeed have a huge impact on a phenotype. For example, sickle cell disease is caused by a single mutation in one base pair. In sickle cell disease, for a given gene, only one base pair is changed or mutated. and that creates a different RNA transcript, which then this codon codes for, in translation, a completely different amino acid. And believe it or not, one amino acid is enough to change a healthy non-sickle red blood cell to a sickle unhealthy red blood cell. So, in general, there are two broad categories of point mutations: single nucleotide pair substitutions and nucleotide insertion and deletions. So, substitutions occur when one base in an mRNA sequence is mutated or is changed into a different. So we replace an A with a G, a C is replaced with a T, something like that. Since more than one codon may code for the same amino acids, these can be silent, meaning they don't alter the phenotype at all. So, for example, this is our normal DNA template, our normal mRNA template, and our normal amino acid sequence. If I change here this GC to an AT, what's going to happen is we're going to create a different mRNA transcript, but because this codon codes for glycine as well, remember, more than one mRNA codon can code for the same amino acid, we end up with the same amino acid sequence. So that's an example of a silent mutation. Sometimes, though, a substitution can result in the creation of a different amino acid. For example, here we have replaced T instead of C. So now we get a codon that is AGC instead of GGC. And this is going to create, or be the codon that codes for serine in translation instead of glycine. And now we have what's called a missense mutation. We've only changed one amino acid in our amino acid sequence. Even more devastatingly, though, sometimes when we substitute or change one nucleotide, it can change our mRNA codon to a stop codon. So instead of it coding for an amino acid, it creates a stop codon. It stops transcription or translation at this point, meaning we lose all of these genes, all these amino acids downstream of that mutation. This is called a nonsense mutation. It's a lot worse because it disrupts all of the amino acids and basically this whole polypeptide after this one point. Second type is insertion and deletions, and like it sounds like this is when we insert an extra or delete a nucleotide from the mRNA transcript. They're often more harmful than substitution mutations because every codon after that point is affected. So, for example, if I insert a U here, right before this ATT sequence, it can change this codon to a stop codon, and we get a missense or nonsense mutation. All of these amino acids are lost. We can also delete out a nucleotide, and that will shift all of the reading frames, all of the codons over one. And it will again change every single amino acid after that point. We call this a frame shift, since it's shifting our codon frames over one this way for deletion or over one this way for insertion. So in general, insertion and deletion mutations have a far greater potential for being harmful to an organism. Lastly, things that cause mutations, called mutagens. Mutations can arise in several different ways. Errors occurring crossing over of DNA replication can result in any of the discussed mutations. Errors in mRNA transcription, translation, processing, all of those can lead to error as well. So, in one example is in chemicals and in radiation, including UV light, can cause DNA mutations. Chemicals and radiation that cause mutations are called mutagens. So for example, this compound, if your DNA is subjected to this compound, it changes the binding site of adenine to look more like that of guanine. So, when replication occurs, we get a substitution of this T to a C. And on the alpha side, when it's replicated again, we get a complete mutation. So now we have a substitution mutation that came from the exposure to this mutagen. In addition, UV light can cause mutations. What I can do is cause bridges between pyrimidines, covalent bonds between pyrimidines to form. So they can be covalently bonded to each other. And these are called dimers. And UV light can cause the formation of these pyrimidine dimers. And they can alter the DNA and eventually cause things like melanomas, like cancers. So these different mutagens can create point mutations and alter our DNA and cause some pretty harmful side effects. Alright, that's it for this section. Please let me know if you have any questions.